Aloha, friends. My name is Avalon Paradea, and I'm the graduate student intern with the South Kohala Coastal Partnership. For today's speaker series, I am pleased to introduce McKenzie, Henrietta Dulai, and Kim Falinski. Casey is a Sea Grant graduate fellow in the Department of Earth Sciences at UH Mānoa, and she is using this research for her master's thesis. Henrietta is also from the Department of Earth Sciences at UH Mānoa, and her expertise is in coastal hydrological processes, groundwater and coastal water quality, and geochemistry. And Kim is a science advisor with the Nature Conservancy, helping on coastal water quality issues throughout the state. Henrietta and Kim are the project PIs and serve as advisors to Casey. And with that, I shall pass things off to our speakers.
Thank you so much. All right, here we go. Thank you everyone for being here today. We will be discussing with you a presentation following the flow, tracing groundwater and nutrient fluxes using chemical tracers in South Kohala, Hawaii. And I'm gonna pass the baton on to Kim real quick to start us off. If I can get it to move. There we go.
Hi, everybody. I'm Kim. I'm based in Honolulu. Thanks for having us here today. The project was started from us talking about how nutrients in wastewater might impact coral reef ecosystems. And I think probably all of you on the call have heard some of the impacts that we think might happen. If there's algal overgrowth, poor coral recruitment, corals that are exposed to high nitrate and inorganic nutrients are more likely to bleach. All of these impacts have been documented by our team and our partners. And so we wanted to really try to understand where the sources of those nutrients and wastewater inputs might be. Next slide, Casey. The overarching question that we were trying to get at, although we have seen many studies that kind of like say, okay, wastewater is impacting here, or we have good groups like Hawai'i Viola trying to go after what the water quality is along the whole coast. With these new advanced tracers that Casey and Henrietta will talk about after me, that we were trying to really pin down source and transport pathways. So really getting to the question of what kind of land use, what kind of practice is transmitting chemicals, and to where on the coast and where are they coming from? So what groundwater path? And I was so excited to work with Casey and Henrietta on this project because these new pathway, these new contaminants of concern, these new ways of tracing the water can really help us get at answering some of the fundamental questions that will be important for management. So with the contaminants that they're going to talk about, we can finally start to pull apart from the difference between landscaping nitrogen, cesspool nitrogen, versus say wastewater treated nitrogen. They all look different with signatures. And so here we come up with this kind of superpower punch of combining all of those things to really understand where the nutrients are coming from and how they're getting there. So which type of thing they're getting there with the goal of them being able to maybe stop the flow of those nutrients and wastewater or landscaping sources before they get into the ocean. So with that, I'll pass it on to Casey.
Thank you, Kim. So ultimately what we did is we proposed the use of a combination of geochemical and pollution tracers, as Kim said, to identify sources of wastewater pollution and their pathways to the ocean. And by analyzing the presence of microcontaminants in submarine groundwater discharge, which I'll talk about a lot, we also refer to it as SGD, we aim to determine the land use categories that may be sources of nutrients and microcontaminants in the SGD. And where they exist, we will confirm a hydrological and chemical connection between different land-based sources of pollutants using the source-specific tracers. So to break that down a little bit more, the target sample sites of our project are centered upon the use of groundwater discharging along the coastline through what many know as coastal springs, but I'll frequently refer to as SGD. And this illustration models the presence of multiple land use categories that may coincide within a single watershed. So these land use categories also cumulatively supply man-made nutrient waste to the groundwater, which enters coastal zones through the SGD. And since multiple sources are present, it could prove challenging to decipher which ones contribute nutrient pollution through this pathway, and hence we'll focus on utilizing the presence of other microcontaminants as our tracers. In the right image is a map of the South Kohala region, where we see the presence of sites that are that exercise large-scale landscaping practices used for using fertilizers and pesticides. And those are visible in the blue squares and the green shading as hotels and golf courses, respectively. And I know it's a little small. I apologize for that. There is also a large quantity of on-site sewage disposal systems, or OSDS, present in the area, which are labeled in purple hued circles. And the map shows our stations that we sampled, where you can see the multicolored circles along the coastline, ranging in red, orange, yellow, green, blue, and purple, right along the coastline. They blend in a bit as we did sample that SGD as close as we could to those major land use categories. And wells sampled are identified in the dark squares upland of the coastline. To help delineate which land use sources could be contributing to nitrate loading, we chose a variety of chemicals to analyze as potential tracers to try to decipher landscaping wastewater from OSDS effluents and potentially leaky sewer infrastructure. And I'll be reviewing our selection of those listed chemicals there at the bottom here shortly. So in addition to the analysis of the microcontaminant tracers, our sampling approach involved water collection for several other types of analyses. Salinity of all samples was recorded in the field and also double checked in the lab after field work to try to verify that we are indeed getting somewhat of a freshwater signal. And dissolved organic and inorganic nutrients were analyzed in the lab, as well as stable isotope analysis of, bear with me, del-O18 and deuterium in water. This is for the purpose of calculating the groundwater recharge elevation. And we also analyzed for del-N15 and del-O18 of nitrate for the assistance in verifying wastewater nitrate contribution. So analyzing these isotopic signatures of nitrate is the classic method. Other research has applied for identifying wastewater, but this method is not foolproof in picking apart all of the wastewater origins. So hence we hope to enhance this method a step further through our microcontaminant analysis and scoring to try to pull apart those mixed signals within the nitrate pollution itself. Cumulatively, we sampled 14 kilometers or roughly 8 miles of coastline. And of that stretch, we have defined 5 general regions, which are outlined in those various colored frames. Our methodology in choosing where we sampled was guided by thermal infrared imagery maps that we attained through previous research. And the thermal imagery shows a slightly cooler temperature signature where the SGD seeps into the warmer adjacent shore water. We focused our sampling efforts on areas with obvious seepage according to the thermal maps and coordinated with our peers at the Nature Conservancy for guidance to these locations. Our friends at TNC were also super helpful in helping us with some local networking for gaining approved access and permissions from the local land managers and sampling purposes as well. So all in all, in our group effort, we were able to sample a total of 45 springs where SGD was present along the coast, as well as 24 inland wells. And then I also wanted to highlight this thermal infrared imagery because it's really fascinating to me. So I've included an enlarged image of the Mauna Kea Resort and Hapuna Beach, paired with an overlay of the thermal imagery, where you can see how those cooler SGD seeps along the coast reveal a thermal gradient signature compared to the surrounding warmer ambient seawater. And ultimately it was in these areas where the temperature signature was that blue to green color, that cooler color, that we were able to locate the springs like the one pictured over on the right. However, not all of our springs were so obvious. Many were concealed throughout rocky shorelines. So here are some pictures of what it looks like to sample SGD in other areas. We targeted springs at low tide, and like those in the top left and center images, while some of select few springs were also much easier to spot, like the ones pouring out in the bottom left. Once we located a spring, we injected a push point rod into the sediment. You can see that in the top left image, and attached the rod to a peristaltic pump pump via Tygon tubing. We were then able to pump the fresh to brackish water directly out of the spring and into sample vials like those pictured in the bottom center image. This enabled us to acquire coastal groundwater samples that were limited in their mixing with the salt water from incoming waves, and wells were much easier to sample, as you can see in the far right images, no need for a push point sampler or a pump. And now that you've seen a little bit about how we sampled and surveyed the coastline for our springs, I'm going to pass it over to Henrietta for a moment, and she's going to talk to you a bit about the nutrients and the stable isotope analysis that we did in the lab.
Thank you, Casey, and thanks everyone again for having us. All right, let's jump into looking at nitrogen, which is our major element of concern here. Nitrate specifically is the most dominant chemical species of nitrogen because of the oxygenated, oxygen-rich nature of groundwater. And then specifically, nitrate comes in different flavors depending on its sources that I will allude to in a minute. But first of all, let's look at our first figure that shows the measured nitrate levels. All you see here are circles representing coastal springs, and ocean levels are highlighted by the dotted line. So those are ambient levels in the ocean. And as you can tell, groundwater, so the coastal springs, bring in all that excess nitrogen that is very much needed by the coastal ecosystems, of course, up to a certain level. Too much of a good thing becomes harmful to them. And not necessarily now discussing what the optimal levels would be, we focused rather on where the nitrogen is coming from. And as Casey already mentioned, we did not always manage to get coastal spring samples that were not diluted by seawater, but those can be corrected for. So the second figure that is named on the x-axis apparent nitrate represents nitrate concentrations that were corrected for dilution by ocean water, which is low in nitrate. And so we like to express nitrate concentrations this way because they really represent, so we can compare oranges to oranges. If all the springs were completely fresh, not diluted by ocean, these are the concentrations that we would see in them. I should also mention throughout the talk, all the symbol colors are matched to the map as illustrated on the left. And the y-axis for all our figures are, well, most of our figures are latitude for easier orientation geographically. So finally, the most right figure on this slide shows that commonly used nitrogen isotope signature in nitrate. And more on that on my next few slides. Before we actually go on to the nitrogen isotope, I would like to mention quickly that we have also collected samples for oxygen and hydrogen isotopes of water itself. As Casey mentioned, one of our objectives was to try to understand the flow path where the water has recharged, what is the land use that it has traveled under, and obviously there will be big differences between water flow paths that have been short and recharged recently under a certain land use versus something that originates very high up in the watershed and gets enriched by a lot of natural nitrogen, for example, from soils. So the left figure here shows all the sampling points, and the right figure shows the actual calculated recharge elevation based on the measured oxygen isotopes. And the recharge elevations were determined in previous studies, such as by Joe Fackrell and Diamond Teixeira. So that's using previous results on the recharge elevation calculation. What the trends show is that we have samples in the southern section that recharge at much higher elevations And on the left figure, you see those lines connecting that recharge elevation to Mauna Kea, although it can be the saddle or Mauna Loa. Nevertheless, the flow path will be to a higher elevation than the samples that are on the north that really would match up much better with Kohala rather than Mauna Kea. We are not insisting these are the correct flow paths, but these are one scenario that could work that would explain those oxygen isotopes observed in groundwater well. And Casey will be using these flow paths then later on to derive land use categories for each point. For now, however, we will use these characteristics of water that the water carries with it, so that oxygen signature in the water, to interpret our nitrogen and nitrate signatures. So my next slide shows something that you may have seen in the past, but if you read about nitrogen source tracking, in that the nitrate, which is a molecule composed of a nitrogen and 3 oxygen atoms, will have different nitrogen and oxygen stable isotope signatures. And on the right, we have kind of a overall global signature one might expect, in that the more manure and wastewater we have, the higher that delta N15 per mil signature will be, so difference, heavier nitrogen isotopes. And as that pool is getting processed more and more in the aquifer, it gets higher and higher in that N signature, but also in the oxygen signature. You also see that there is quite a crowding of different categories at the bottom left corner, where we can have overlapping signatures coming from soil, fertilizers, and precipitation in certain cases. So our samples are plotted on the left figure, where clearly, and again, based on past studies, it has been shown that nitrogen signatures, delta N15 of higher than 7 are most likely of wastewater origin, being it leaky cesspools or treated effluent or leaky sewer lines. And so the story goes that once waste is added to the aquifer, so that's organic matter, and that organic matter is then being processed by microbes. They do the good job of breaking down all the organics, but at the same time, they chop off the ammonium from that organic molecule, and that ammonium then gets converted to nitrate thanks to, again, some other groups of microbes and the oxygen that's present in the aquifer. And the fate of that nitrogen can be further followed oftentimes as it gets further processed by denitrifiers, which then again change its isotopic signature. So it's not just what was released to the aquifer, but how all that signature got converted by microbes. And that's a story we are not able to put together necessarily clearly for our flow paths. And so the question that stays still relates to everything that's below that wastewater signature on our left figure for all our samples that are less than 7 per mil of delta N15. Do they have wastewater, or are they all fertilizer or natural nitrogen? So certainly for a few samples on here, we can tell that they are carrying that wastewater signature, but how about those others? So on my next slide, we have, okay, so, yeah, thank you. We now have a different view of these isotopes. Specifically, we draw from that water characteristic, so that oxygen signature that the water carries, we said it's very different along the Kohala coastline. As that ammonium is chopped off from that organic molecule and is being nitrified, so being converted to nitrate, microbes grab one of the oxygens from the atmosphere. So I'm including that here in black in my equation that signifies the nitrate oxygen composition. And the microbes take 2 oxygens for that nitrate from the water itself. And so now, if we transition into the figure on the right, we now have plotted oxygen signatures in the water on the x-axis and oxygen signature in that nitrate on the right axis. And if indeed one of the oxygens come from the atmosphere or dissolved gas in the water, and 2 oxygens from the water itself, and we plot an expected theoretical line, which is the black line on this figure, we see that most of our samples align on that, which means that most of those were actually produced within the aquifer. And what would make sense, one possible scenario is that all the wastewater that was released and the ammonium was released from it was then nitrified using that oxygen within the aquifer. So it was not released as nitrate, but as ammonium or other organic nitrogen. So this puts a very, very strong evidence for that wastewater source and also nitrification and then further denitrification within the aquifer itself. Another source how one could produce points that plot about this line is fertilizers. And you see that, sure, there is possible fertilizer sources, but most would be really from that wastewater. What I hoped to illustrate with this explanation on these nitrogen isotopes and the nitrate is that although we definitely see wastewater signature in many of the samples, not all can be explained just by wastewater, and that we don't have a really clear-cut answer for all our questions. And so that exactly what's led us to exploring these other tracers, specifically microcontaminants that Casey is now going to talk about.
Thank you, Henrietta. So as we can see, it can be a bit tricky identifying the exact sources of wastewater contributing to the groundwater nitrate accumulation. So we chose a variety of microcontaminants to analyze as possible tracers for the different sources. Overall, 14 potential tracers were chosen and analyzed in all of the samples, but 10 of the 14 chemicals showed positive results in 20% or more of the samples, and these are featured in the table. So we excluded the 4 microcontaminants that yielded no results out of the table to save a little bit of space today. Originally, we had selected 3 chemicals as leaky sewer tracers, but only 1 yielded positive results, which was BPA. We chose BPA, highlighted in blue, as a possible tracer for this specific land use due to its abundant use in plastics and resins. For our landscaping tracers, which are highlighted in green on the left, we targeted chemicals common in restricted use pesticides with known sales records in Hawai'i County, including ivermectins, carbamates, and pyrethroids. But we also included analysis of glyphosate in our landscaping category of microcontaminants, since it is common use as an unrestricted herbicide found in Roundup. And for tracing OSDS, we analyzed water for some common use pharmaceuticals, which are highlighted in red and include caffeine, carbamazepine, ibuprofen, and sulfamethoxazole. Chemical characteristics are important in selecting these tracers. Most of these microcontaminants photodegrade rather quickly, so but they have a relatively short half-life in water, but also longer half-lives in soil. And this made them ideal tracers in SGD for our land use categories, considering that they have a relatively longer, they last a bit longer in the soil. And then of the 10 tracers that we analyzed with positive results, 3 of them denoted with asterisks on the right of their names, ivermectins, carbamates, and sulfamethoxazole. Those were present in 100% of our sample sites, SGD and wells included. So here are the results from 5 of our microcontaminant tracers. And I know this is a pretty busy slide, so I'm going to try to just break down some highlights here where we noticed a couple of preliminary trends. Like Henrietta said, our y-axis is the latitude of the location of our samples, and the x-axis is going to be the concentrations of found in those particular microcontaminants. And you might notice a little white triangle at the bottom, and that's just to denote our detection, our lower detection limits for the microcontaminants. I believe the pyrethroids one is off, and I need to correct that. It should be closer to 0. But let's look at the trends here for a moment to move along. So we did notice that in most cases, the microcontaminants present were measured in higher concentrations along the coast than they were in the upland wells. And this indicates that these microcontaminants are reaching the groundwater through applications occurring where the land use is denser and prevalent along the coast, as opposed to the more rural landscape where the wells are located. And as I mentioned earlier, only one of our selected chemicals intended for tracing leaky sewers yielded results, which was the BPA, and is located all the way in the left figure, the far left figure. Overall, we see little to no BPA in our SGD. However, it is most present in 25% of our well samples. We also see that in our green hued sample points across all of our figures here, those stations were sampled in the community, in the Puako community area, where private residences make up the majority of the land use. So you may notice that the pharmaceutical presence, as you see in the center images highlighted in the red-ish square, is relative. Those pharmaceuticals are relatively common there, as opposed to other areas where OSDS was not as common. There are similar trends as well amongst the data for measured restricted use pesticides. Pyrethroids in particular appears present most commonly near large scale landscaping practices, but also had a few pretty positive hits over in the Puako community as well, which it could possibly be due to application by private landscapers. But like I said, because it's a restricted use pesticide, we have to keep our minds open as to how the applications were brought there. And avermectin was present in 100% of our samples. Although avermectin is an RUP used as an ant insecticide, other similar chemicals within the avermectin family are used for various other reasons, especially as an anti-parasitic in human and livestock pharmaceuticals, which could be contributing to its abundance detected through all the sample sites that when we did our lab analysis. So after the completion of our laboratory analysis, we assigned a score to each microcontaminant chemical based upon their individual likelihood to be present in SGD, and as a result of their application with individual land uses. For chemicals considered unlikely to occur in wastewater relative to a land use category, those were scored as a 0. And while those chemicals with a known usage as described here in this figure, we assigned them a score of 1, concluding that in our scoring calculations, a 1 indicates the assumed presence due to land use application, and a 0 implies that the chemical is not related to the corresponding land use. And in these calculations, we were able to essentially score how the prevalence of those particular microcontaminants correlated with the land use categories that we are trying to delineate nitrate waste from. I'm going to hand it back over to Henrietta for more on the pollution scoring.
And you could tell that we don't really have a golden bullet to say, okay, if I see this chemical, it definitely must be a sewer leak or necessarily a certain type of landscaping. So there is still some ambiguity in these pollution scores, but I think we tried our best to select and weed out which ones would really be relevant. And for example, a leaky sewer line score will be composed of, as you've seen on the previous table, not just BPA, but a little bit of carbamazepine and some other chemicals that might be present even in a more processed wastewater. We also then have the OSDS scores, and the OSDS scores that should include cesspools and septic systems would be the pharmaceuticals that would degrade, but we find them. So therefore, we must be seeing some really raw sewage coming through. And then for the landscaping score, again, those are those restricted use pesticides and herbicides and so forth. And the pollution scores really are kind of arbitrary in a sense that just because we see a number closer to 1, which gives us high confidence that there is chemical connection, and that is the one primary use of this pollution score, they are not all equal, meaning if I see a sewer score, that means I see a high sewer score, so closer to 1, that means I found a chemical that suggests that there is leakage from sewer, and so I'm pretty high, I have pretty high confidence that it's in there. I cannot yet, however, tell how much nitrogen is that corresponding to. I can just tell that there is for sure a chemical connection. Same for the OSDS scores. Again, the higher the pollution score, the more confident I am that I'm seeing chemicals that are coming from raw sewage. And the same for the landscaping score. And those repeating patterns, for example, all the purple dots, all the red dots, indicate that pretty much what this boils down to, that wherever we have wastewater, we also are likely to see those landscaping scores getting high just because there are people. Therefore, there is some land use happening as well. But I guess for now, we are still processing these pollution scores. We are still fine-tuning them. So Casey is going to defend in a couple of months, and we'll be working more to fine-tune these. But for now, we can tell that there is chemical connection between those land use categories that we have picked. And we have a suite of chemicals that can tell us that that chemical connection exists. And our next step also is then to further use these pollution scores to then assign to different land uses certain weights to derive the contributed nitrogen from each. And so our conclusions from these studies so far is that nitrogen was found in excess of ambient levels in several coastal springs. And where that nitrogen is coming from was, of course, the big question here. We used nitrogen isotopes of nitrate and oxygen isotopes of nitrate that show that the majority of that nitrogen might be originating from wastewater. Indeed, that has been confirmed by us finding pharmaceuticals in the coastal springs. But some of the nitrogen may be coming from landscaping practices, so using fertilizers that are used along with those pesticides and herbicides that we have also found in the coastal springs. And so for the next steps, as I mentioned, Casey will be working on land uses, deriving what type of land use exists along those flow paths that we identified based on the oxygen isotopes, and also fine-tuning which chemicals best represent which pollution scores, and then also allocating nitrogen releases to those land uses. So on the next slide, we have these final next steps indicated in, as I already mentioned, analyzing more of these results further for including wells, how much nitrogen was added in between. And I guess I pass it on to Kim to discuss this a little bit more.
No, I think you did a great job. Hold on, let me see.
I think I stole Kim's slide. My apologies.
No, that's okay. I'm just wondering where the lag was on the Google slides. I just want to thank Henrietta and Casey for working so hard to help you guys understand all those different contaminants of concern and maybe run back and summarize the great work that they just presented there. For the first time, we can see probable flow paths from which sites are being contributed from Kohala versus Mauna Kea. So that's the first thing, is that those del 15, del 18 ratios can really help us figure out where the water is groundwater recharging from. So that can help us answer one of those questions. The second thing that Henrietta described was how the nitrogen isotopes alone are often really mixed. And so you get kind of numbers that are really ambiguous. And she was talking about how that ambiguity makes it so you can't have like the solid like hammer that you need to be like, okay, it's this. But that the added layer of all of those different pharmaceutical compounds can really contribute to figuring out, okay, which sources are from which place. By doing this along that big coastline, we're able to finally say, okay, maybe we have areas that are hotspots that we hadn't considered before. And I really look forward personally to taking this to the even next step of comparing it to the coral reef layers, the coral bleaching layers, and some of the other kind of metadata we have biologically in the ocean to see if there's connectivity there. So it's one of the next steps, I think, on my side, is to start eventually piecing those data sets together, along with working with the managers to kind of identify, you know, maybe there are chemicals, we only work with the chemicals we could know about, but it's possible also that there are other different types of contaminants that once we start working with managers, we might be able to more precisely try to pull apart where in the system some of these contaminants are coming from and look towards fixing the sources. I can't say enough that I'm trying to understand where the Hawai'i is coming from and how it's mutating as it goes along the coast is really important for us to being able to really try to improve our water quality. And I'm really grateful to Casey and Henrietta for bringing kind of the hammers of these new tools to bear on those questions. And I want to thank you for your time, and we're open to questions from all of you guys on this pretty technical presentation. So thank you.
Mahalo, everybody. That was an amazing presentation. And so I see that we have a couple of questions in our chat. I'm going to go ahead and read these off in order. But folks, feel free also to maybe raise your hands or type more questions into the chat. So the first of these is from Becca. She was curious. She asked a question about the BPA results, since these are upland wells. Is it safe to assume maybe the BPA came from the piping of the wells, or were these from sites near the sewers?
That's a question I've pondered myself, Becca, is that perhaps it could be a part of the infrastructure of the wells. But we have not yet explored verifying that, so to say.
Thanks.
Yeah. Good question.
Awesome. Mahalo for that, Casey. Another question from Rachel Mason is, for the chemicals you didn't detect, has it been established that they are easily transported in groundwater? Might they absorb onto soil or rock on the way to the ocean or something along those lines?
Sorry, could you repeat that one more time? It broke up a little bit.
Oh, sorry about that.
No, you're fine.
Yeah, so Rachel asked, for the chemicals you did not detect, has it been established that they are easily transported in groundwater? Might they absorb onto rock or soil on the way to the ocean or something like that?
In terms of the microcontaminants?
Yeah, probably.
I'm sorry, it keeps breaking up. I don't know if it's just
okay. I can maybe answer that. All these microcontaminants we have looked at are organic molecules, and therefore they will most likely be sorbing onto surfaces. In fact, when we sample, we take good care of, using the right protocols. And so what gives us confidence in that there is not huge retardation, in that some of these really have very short half-lives. So if we see them, that means they had to propagate through the aquifer. And I would use, for example, ibuprofen as an example. Although groundwater is dark, so no photodegradation happens, there is still oxygen for the microbes to use, so it breaks down relatively quickly, and we still see it in the spring. So even though you are right that these chemicals might be retarded within the aquifer, soils and rock surfaces, it's not 100% absorption. There is still some propagation down through the flow path and then appearing in these coastal springs. And that is one reason also why we cannot use these chemicals, as in if I see X grams, that means that how many gallons of wastewater were released, because we might not be getting everything down at the coastal spring due to this retardation and biodegradation. But if we see it, then we are at least certain of that chemical connection. And so kind of we ignore the fact that they are not necessarily conservative.
Wonderful, thank you, Henrietta. Can you folks hear me okay right now?
Yeah, you're coming in fine on my end.
Okay, I wanted to switch out of the headset because sometimes it's a little touchy. So another question from Rachel is, are any of these chemicals likely to have effects on ocean life at the concentrations you detected after dilution by ocean water?
Casey, could you move a slide one over, because I just inserted a figure that we were thinking about, considering about including forward, not back. Yeah.
Or down,
One farther.
up further.
Not back, but forward. Slide 22.
It's not, let me see. It's making it end right here because those ones are covered up for now. Let me see if I can edit them on my.
I just added 22 here. Anyway.
Henrietta, for whatever reason, I wasn't able to update the presentation live either, so I'm not sure what's going on.
Okay. Maybe I can share my screen then.
Okay. Sorry about that.
No, my bad here. We were actually thinking about including this figure, and we anticipated this question, but we thought we already provided so much information. So definitely there is great concern about these chemicals being present in the environment. And I must say, none of these concentrations have passed the acute harmful levels for aquatic ecosystems. However, the research is still out there to study the chronic exposure of organisms to these chemicals. And indeed, if you think about that coastline, the coastal ecosystem that's day-to-day continuously being bathed for years in a slew of chemicals, obviously, they have effects that we really need to look into. But there have been studies elsewhere, not in Hawaii, in seeing how these chemicals affect certain organisms. And so we picked whatever we could for marine organisms and found what's called a no effect concentrations, which means what is the highest concentration of these chemicals that will still cause no harmful effect to the organism. And when you take that and use it, and you divide your actual measured concentration by these no-cost effect concentrations, then you get this number, which really is a multiple of how much higher you are from that no effect concentration. And we call that a risk quotient. So what's clear on this here, we have risk quotient plotted on the x-axis, is that caffeine and ibuprofen and some of the SMX, so some of the sulfamethoxazole, which is antibiotics, are higher than 1, which means they represent a high risk for the coastal ecosystem. And everything that's below 1, and on this image you can't really tell because some of the risk quotients for the caffeine and ibuprofen are so high that the 1 is hiding down here. But some of the green, and definitely most of the triangles and the crosses, are actually providing an environment that may be harmful for the coastal ecosystems. So I must point out that this study really focused on just like seeing what are the concentration levels and how we can use them as tracers, not how they affect the coastal ecosystem. That's the next step. Now that we are confident that we are seeing that chemical connection, the next level is then for people to go in and really study the effect of those for Hawaiian ecosystem, Hawaii reef specific organisms. So that's our call to the scientists and communities out there is to try to understand these effects.
That's wonderful, Henrietta. I really appreciate that you had that to look at, because I had a similar question to Rachel, which was, how harmful these microcontaminants might be to marine life. So obviously a lot of work still to be done there. Becca mentions it's interesting to see how high all of the pollution scores are for the Waikoloa region. Wonder if a good next step is to share with the managers there to see if there's anything that can be done to reduce inputs to the shoreline. And I definitely have to agree with that statement. Any thoughts on that?
I can just say that I look forward to Becca's support in reaching out to the Waikoloa managers and maybe taking that particular part of the data set and sharing them with them specifically, and figuring out the best way to communicate this information. So yes, I agree. There's even little blips up in Kauai High that I find interesting. It wasn't just Waikoloa that kind of stood out. The focus has been on Puako for so long, and so it's interesting to see some of the other areas thrown into the same mix.
Yeah, definitely. I live in Waikoloa Village, so I would like to see our pollution and contamination reduced for a lot of reasons, obviously. And then Elliot has a question as well. He says, I know this is outside of the study, but any thought about how sea level rise may impact nutrient pollution? And could this have cascading of impacts on coral reefs?
I can definitely address that. We have done studies already where we contrasted spring tide, neap tide cycles, and even king tides to look at water quality along the shoreline. So this was done on Oahu, not in West Hawaii, but the same story would be applying here, that unfortunately, a lot of the cesspools and even the sewer lines are already at groundwater level. And with every spring or king tide, high tide, groundwater level is lifted within the subsurface with those tides, pretty much flooding even the well-functioning septic systems may act as just cesspools if they are flooded. So we have demonstrated that we see a difference in the chemical composition in the groundwater and coastal springs during those high tide events. And we were kind of hoping to contrast things, and then maybe that king tides would be showing this much more clearly. But it turns out spring tides are already bad in certain regions. So we looked at Kahalu'u on Oahu and Waikiki, and both sides already have big problems in that groundwater inundation of wastewater infrastructure. And it's not just cesspools that we blame, it's also sewer lines. You see these chemicals showing up in storm drains as well. There are regions in Honolulu, Mapunapuna region, for example, where with every spring tide, the streets flood due to, again, groundwater inundation. So this is not overtopping of the ocean. This is the groundwater surfacing in low-lying areas. And we see these chemicals in that water as well that floods the streets. And so this then goes beyond the ecosystem health problem. This is now human health, because cars, people are wading through that water. And then of course, eventually it flushes into the coastal zone. So yes, that question is very, very important and must be followed up. And the state is aware of this. And so action must be taken in making our wastewater infrastructure more resilient to sea level rise.
Mahalo, Henrietta. I don't see any other questions immediately in the chat. Lillian did share an interesting paper on, let's see here, chemicals on coral health. So that's interesting. Mahalo for sharing that, Lillian. I do have one quick question, which is in regards to water sources. I saw that in that slide where you describe water sources, Waikoloa water stems from Mauna Kea. And I'm just curious, you know, I recently read the draft EIS that the Army put out for 23,000 acres of state leased land up at the Pahuakoloa region, and there are aquifers under that land. Is there any thought on sharing this information with the Army, or any work done to look at contaminants that could be coming from that particular the aquifers underneath the Pahuakoloa region? Or is it just a broad concept that water in Waikoloa, for example, comes from Mauna Kea with no exact source point?
Water sourcing is tricky. As I said, I guess I hope I was clear. My preface is I plotted those flow paths up to Mauna Kea, but actually Mauna Loa is the kind of the flank that leads and feeds this aquifer or assumed to be. So there are questions about that. So we will be following up more on that. But I would say we would love to share the results with as many folks and groups as possible, with bearing in mind that there is obviously sensitive information in this for the different communities that are part of this data set. And so we will be looking towards TNC in guiding us, hopefully, and helping us on how best to navigate sharing this among the different community groups.
Wonderful. Mahalo, Henrietta. Do we have any other questions from people? I'm not seeing anybody jumping out right now. So if that might be it, I just want to give a big mahalo to all three of you ladies. I really appreciated this conversation, and looking forward to seeing where this research leads in the future as well.
Thank you so much for the opportunity.
Thank you very much.